this situation. Thank you all for joining today's webinar. Please confirm me. Principal sir is there?
Yes, I am here.
Yes,
Madam Salma, I am here.
Okay, sir. Thank you, sir. We're just about to begin our today's program, International Webinar on Modern Approaches and Techniques for Advancement of Molecular Biology. First of all, I would like to invite Dr. Gautam Chatterjee, Principal, Krishna Chandra College, to say few words. Sir, please.
Good morning to all of you. A very good morning. Actually, I want to express my sincere thanks to our respected chief guest, Dr. Shobhan Roy, and our respected research person, Professor Subhas Chandra Lakhotia. Welcome, sir, both of you. We are very much pleased for your giving time from your busy schedule. It is my great pleasure to welcome you here this morning to this international webinar. It is a great pleasure and excitement that we have within us such very great resource persons of WHO. Thanks to Department of Biotechnology for their kind cooperation and for their kind consideration to our colleges. Actually, we are very small college in a village. We have some hindrance and limitations. But besides this, we try our best. Zoology department, it is a very good department with a good faculty member. They are doing their work very well. Though we have overcome our various limitations, I pray the support of all of you. So, I do not longer my talk. Hope that this webinar will be successful in fostering better communication and interaction among the members present over here. Again, welcome to all of you in our international webinar. Thank you, everybody.
Thank you, sir. Thank you for your nice words. Now, I welcome with respect our honorable chief guest. I am extremely privileged to introduce Dr. Shobhan Roy, Senior Scientific Officer and ex-officio Senior Scientist, Government of West Bengal, Department of Science and Technology and Biotechnology. Now, I would like to request Dr. Shobhan Roy to convey his valuable words for this webinar. Sir, please.
Thank you. Thank you very much. Very good morning. The Honorable Professor Subhas Chandra Lakhotia and all other dignitaries, competent dignitaries. I dare to thank Dr. Gautam Chatterjee, Principal of the Krishna Chandra College, and Dr. Jt Mukherjee, convener of this webinar, for inviting me to this high-end event happening in your veterinary institution. I especially thank Professor Lakhotia, whom I am seeing after 28 years when I left Banaras University. I thank him for speaking at this event organized by a less known but nearly 125 years old college situated at a remote locality of West Bengal. This college has served silently for diffusion and diffusion of education in the local society, generation after generation. There is 4 times increase in the world population during the last 100 years. This will climb up to 9.5 billion by 2050. By and large, 2 billion population belongs to South Asia, only South Asia, and India contributes near about 1.4 billion. 15 to 20% of world population suffers from malnutrition. Poverty has so vehemently transcended the imaginable reality that many penniless poors die directly or indirectly out of hunger without record. Although reasons have multifaceted explanations, yet multiple major breakthroughs must be achieved in future in the milieu of foodstuff production, health, medicine, and other dimensions of welfare activities. An urgent and well-thought approach towards the utilization of agricultural biotechnology, and particularly lead towards the transgenic developments, may be essential, although it is still now controversial. I am not going into this dispute, but there is no ambiguity that past research at molecular level has brought revolutionary changes in most of the biological sectors of human welfare. All these things are, however, known to you. I shall like to point out here some essential thrust areas that I need revisits must be made at the present scenario. Firstly, agro-resource management, crop diversification, and imparting the concept of balanced food to the society, and particularly to the downtrodden society. Exploring innovative research capabilities and engagement of more competent resource persons in science education honestly. Training towards startup entrepreneurship is very much essential. Minimization of bureaucratic dogma in the science sector. And very important to me, to come out from the existing concept of publication values, to come out of the existing concept of publication values in the name of so-called impact factor, and to bring attitudinal changes among scientists to publish more and more in Indian traditional veteran journals. I feel what you have written is more important than in which journal you have published. Let the learned readers decide about the standard of your publication, standard of your paper, and not merely the editor and two reviewers chosen by him. It is time to create self-reliant India. It is time to create self-reliant India and rethink on global gradual phagocytosis of our journals by European and American brands. You have to rethink that, am I delivering the required output to the society, or I am working just for my self-career enhancement? Don't allow statistics to dictate the biological output. Rather, statistics provide best possible judgment, which may not be always adequate to explain the minute feelings and sense of the nature. Last but not the least, whatever stage science attains, ZPGR or zero population growth rate is essential for developing nations. This has already been achieved by a number of developed nations. Hunger and poverty, exploitation of resources shall never be eradicated if this cannot be achieved. Resources of limited supply, demand is quality dependent, contagiousness is increasing, and consequently competition is also increasing with more and more contagiousness, more and more population rise. Demand for the tricks to survive, plus human greed and lack of charity, including domination of medieval values, are pushing our society to further instability. And with this, and with thanks to all of you, I again thank you once again for your patience and valuable time. I wish all success to this international webinar. Thank you very much.
Thank you, sir. Thank you for your inspiring words, and thank you for giving us your precious time. Now, finally, I request Dr. Jt Mukherjee, convener of this international webinar and head of the department, Krishna Chandra College, to say her words.
Am I audible?
Yeah, you are audible.
Thank you, Dr. Salma Khatun. A very good morning to the Honorable Keynote Speaker of the webinar, Professor Lakhotia, Dr. Gautam Chatterjee, Principal of Krishna Chandra College, Dr. Shobhan Roy, Chief Guest of the program, and all the faculty members and the students present here. It is the honor of our college to welcome you all. This webinar is dedicated to bring together a scientific community, including the faculty members, researchers, and students, to strengthen the implementation of science research agenda to address key barriers and challenges in the field of genetics and molecular biology. This event will run for 3 days, one lecture each day. In this new normal condition, this webinar is an attempt to accelerate our knowledge in the field of molecular biology with modern aspects. We have Professor Subhas Lakhotia, pioneering scientist in the field of cutting-edge research in genetics, as the keynote speaker. Dr. Sumetha Roy and Dr. Sujoy Ghosh, our two other invited speakers, are the distinguished scientists in their own field of research. This is the opportunity for the audience, especially the students, to learn from the leading experts in the field of modern approaches in biology. The purpose of this webinar is to facilitate discussions among the researchers and coordination with students who will be budding scientists in near future. This platform mainly aims the scientific community of biology to disseminate knowledge and to amplify and strengthen research. Over to you, Dr. Khatun.
Thank you, Dr. Jt Mukherjee. Without further delay, we would like to start our technical session. Now, I request again Dr. Jt Mukherjee, Assistant Professor, Department of Zoology, Krishna Chandra College, to introduce our today's keynote speaker.
Okay, thank you so much again. Okay, so today we have Professor Subhas Lakhotia as the keynote speaker for this webinar on modern approaches and techniques for advancement of molecular biology. We are pleased that Professor Lakhotia has kindly agreed to deliver his lecture in this occasion. It is my privilege to introduce Professor Lakhotia in front of you all. Actually, Professor Lakhotia is one who does not need an introduction and one of the most distinguished scientists this country has ever known. Professor Lakhotia is an eminent cytogeneticist, academician, distinguished professor of zoology, and INSA senior scientist at Banaras Hindu University. Professor Lakhotia started his early career in Kolkata. He graduated from Vidyasagar College in Zoology Honours in 1964 and obtained his master's degree in Zoology and Comparative Anatomy in 1966 from University of Calcutta. He completed his PhD from the same university in 1970 and did his postdoctoral studies at Delhi University during 1970 to 71 and started his career as a lecturer in Bardhaman University in 1971. Then he moved to Gujarat University in 1972 and joined Banaras Hindu University as a reader in 1976. He became a full professor in 1984. He carried out advanced studies at Institute of Animal Genetics of the University of Edinburgh during 1972-73, at the University of California, Irvine during 1984-85, and at Massachusetts Institute of Technology, Cambridge in 1985. He has been Emeritus Professor in BHU from 2011 to 2015. He continued his academic activities as INSA Senior Scientist from 2017 to 2019 at the Banaras Hindu University, where he is now working as lifelong distinguished professor and SERB distinguished fellow. Professor Lakhotia has carried out his research on the developmental rules and regulations of the non-coding HSR omega, heat shock, and a few other novel genes in Drosophila using genetic, cell biological, molecular, and recombinant DNA approaches. Roles of these genes in modulating apoptosis and neurodegeneration in Drosophila models of human diseases and cellular effects of certain Ayurvedic formulations using flying model are the primary focus of his research. He has more than 180 publications, including original research papers, review articles, and book chapters. He has also written many articles relating policies about higher education, research publications, their assessment, and ethics. He has already completed 33 specific research projects funded by various national agencies like UGC, INSA, DAE, CSIR, DST, and DBT. Has already mentored more than 30 scholars in their doctoral studies. He has been member of editorial boards of a number of national and international research journals. He is also member of 10 organizations of national and international repute. Professor Lakhotia has immense contribution to the establishment of the Department of Molecular and Human Genetics at Banaras Hindu University. Professor Lakhotia has received the Young Scientist Medal of the Indian National Academy 1975 and a Career Award in Sciences of the University Grants Commission of India in 1979. The Council of Scientific and Industrial Research awarded him the Shanti Swarup Bhatnagar Prize, one of the highest Indian science awards, in 1989. The same year that he was selected as a National Lecturer by UGC. The Commission honored him again in 1998 with the J.C. Bose Award, and he received Insa Sundarlal Hora Medal in 2002. The Banaras Hindu University gave Professor C.N.R. Rao Education Foundation Award in 2007. Professor Lakhotia has held the Ramanna Fellowship of the DST Government of India from 2009 to 2012 and the Raja Ramanna Fellowship of the Department of Atomic Energy from 2011 to 2015. He held the Jawaharlal Nehru Birth Centenary Fellowship and Senior Scientist position of the Indian National Science Academy in 2009. He was elected as a fellow by the Indian National Science Academy in 1993, by the Indian Academy of Sciences in 1994, and by the National Academy of Sciences India in 2002. He was elected the Senior Fellow of the Cells Trust Society International in 2017. Thank you so much, sir, for joining and bestowing us with your own presence. Please join me in welcoming Professor Subhas Chandra Lakhotia. Right now, I request everyone to mute yourself during the talk. The screen is all yours, sir.
Thank you. Thank you so much. I'll start my presentation. Is it visible now?
No, sir, it's not visible.
No? No?
No, sir.
What's wrong again? Is it visible now?
Yes, sir.
Okay, good.
But the slides are not there, sir.
The slide is there.
Principal sir is there. Sir, the slides are not there.
Not there.
So it's showing you are presenting.
Yeah, but my slides will be visible also. So I'll start again.
Okay, sir.
Should I do entire screen or window?
You have to select entire screen.
Entire screen, sir.
Yes, sir.
All right. Is it okay now? Slide is visible?
No, sir, it's not visible.
So, No?
Yes, it is visible.
Okay.
Yes,
Okay.
okay.
I hope it
It's
won't.
now in full screen. It's
Good. I'm so happy to be talking to you all, and I thank the college principal, the West Bengal DST, Dr. Roy, and the department for organizing this meeting and asking me to talk about this. What I've decided to do is not to talk about research, but just to let students and faculty know that how we can use Drosophila for many different kinds of experimental studies in biological sciences, not just genetics and so on. Before I go into those details, I'll just share, let the students and others know my research interests. As I mentioned, that one of the main interests is on the non-coding HSR omega gene in development, thermotolerance, and so on. And then we are also looking at stress proteins in tumor development and Ayurvedic biology to understand the molecular and cell biological studies on effects of some Ayurvedic formulations. All studies have been done in Drosophila. And of course, as was also mentioned, I'm quite passionate about higher education and research assessment policies, and I've been continuously writing on these issues. And as Dr. Roy pointed out, I definitely would encourage that we should worry about impact factor. We should worry about the quality of publication, not the cover of the publication, not the journal cover. But that's a different issue. All my publications are available at SERB. Coming to the main topic, what is important now is that as we have improving in our biological sciences, our teaching has suffered substantially for a variety of reasons. Infrastructure missing, the policies on animal rights, and so on and so forth. And consequently, most of the life science courses are without life now. The practicals are not held. Of course, the pandemic is now a different situation, but even otherwise, we have been suffering on this. And that's why we thought that maybe Drosophila can be a good experimental model for undergraduate and postgraduate laboratories. And I'll just try to explain some simple experiment that can be done in most colleges and most university departments. Now why is this necessary? Experimental studies are essential learning tools for students of biology. Not only that, they provide better understanding and appreciation of what is learned through lectures and reading. Hands-on experience and expertise arouse curiosity, and that's an important point, that only when students do things with their own hand, they see doing it, they become curious, and that's where many times important discoveries are made, and it makes them good future scientists. And since biological sciences have undergone tremendous progress in the last 40-50 years, absolutely unbelievable kind of progress, the domain of experimental work has also substantially expanded. Today we want to put emphasis on molecular biology, but let's not forget that biology is not just molecular biology. Biology is biology, and we need to understand all aspects. And therefore, laboratory work with live animals which is not now getting the due share, should be revived. Not just dissections and so on, but many kinds of experiments that we can do and understand biological processes in that process. And therefore, and since what we learned in biology, that biological processes in all across the living organisms are fairly common. What we learn from bacteria, what we learn in plants, what we learn in insects, what we learn in humans, what we learn in fishes, they all have a common basis, and that basic unity is what helps us to use different model systems. And Drosophila is a wonderful model for experimental studies, because one important point for my present emphasis is it's not affected by restrictions on animal use. There is no restriction how many flies you can kill or how many flies you can dissect and so on and so. And it's really a beautiful system to work with. That's what I'll try to explain in the next 45 minutes or so, that how we can make use of it. So Drosophila should be a good model, and we are trying to propagate that. I'll come back to that point later on. Now this is where the fly started. Thomas and Morgan, the father of modern genetics, if we can say, if Mendel was father of genetics, Thomas and Morgan has been the father of genetics. When he started using Drosophila in 1908 or 1909, and set up his fly room in Columbia University Zoology Department with some great names. He himself, of course, being the chair, of the department and lab, Sturtevant, Jt Muller, C.B. Bridges, and many others. You name any great geneticist or molecular biologists today, they all will trace back to Morgan's academic lineage. And this has been a wonderful place that fly to, where lots of things happen. And in India also, we can trace back our academic lineage to Morgan. For example, one of the earliest Drosophila researcher in India was Professor S.P. Chaudhary, who started his career in Calcutta University. He was trained for PhD with Jt Mukherjee, who himself was a student of Morgan. Then Professor Chaudhary trained Dr. S. Mukherjee, with whom I got my PhD. Dr. S. Mukherjee did his PhD with Kurt Stern, who himself was a postdoc fellow of Thomas Morgan. And of course, Professor Chaudhary was my examiner in MSc as well as PhD, so I have two lineages from him. But then the other great names that started Drosophila work in India in the 1960s and 70s, Obaid Siddiqui. He was a student, he learned animal behavioral genetics with Samuel Bunzer, who was a student of Max Delbruck and who had been trained by Sturtevant. And of course, Pontecorvo, with whom Obaid Siddiqui did his PhD to begin with in fungal genetics, was a student of Muller. Then, so likewise, you see, the other important school of fly genetics in India from dating back from very early is in Shetty and then Professor Krishnamurti. They all again trace back to Muller or Dobzhansky, and you trace them back again to Muller, to Morgan. And Professor Krishnamurti, student Raghunath, who has been there and continues to be active, like me. And so you see, what this, I'm just trying to say that now we have in India about 150 Drosophila researchers, starting from about 4 or 5 till about 2000. Now it has expanded, and we have enormous resources available in the country to make use of this organism in classroom studies as well. And this is, of course, its life cycle. It completes the life cycle in 10 days' time. The eggs hatch in about 24 hours. The eggs which are laid hatch in 24 hours. First instar, 24 hours. Second instar, 24 hours. Third instar, 48 hours. Then they go to stationary phase of pupation. And after 4 or 5 days, the fly emerges from the pupa. The fresh fly, young fly, which are very soft, and then within 4 or 5 hours, these flies are ready to mate. And then they live out for about 2 months or so, or 2.5 months, and then the cycle continues. So the important point is that within 10 to 15 days you get next generation, and it lasts longer. And that has been the major advantage why Morgan started using this for the genetic studies. And of course, this is again a picture from Morgan's lab. Within few years, the first mutation, of course, that Morgan obtained was the white-type mutation in 1909, which actually changed his own philosophy, his own understanding about the chromosome base of inheritance and natural selection. And that's where one thing that we learned from Morgan is, to begin with, when he started work with fly, he didn't believe in chromosome base of inheritance. He was not sure of Mendel's laws of inheritance, and he didn't believe in the natural selection small mutations. But then, within 3-4 years, he with his students like Bridges and Altenberg and Muller completely reversed his ideas. And this is an important point to understand in science, that we cannot remain dogmatic. We can accept that we were wrong, and that's what Morgan did. And within few years, he identified numerous mutants, started mapping them, and so the fly became what we now call as Cinderella of genetics, one of the most lovely organisms that we have. And of course, now in classroom studies, some of you, if you have done that, we use various side mutants, wing mutations, body color mutants to look at laws of inheritance, Mendel's laws, first independent assortment, segregation, linkage, crossing over, and so on and so forth. And these talks are available. Now we are trying to create facilities that if colleges and university departments want these flies, we can provide them. Only thing, they will have to maintain them for 2-3 months when they are in the classroom. But we will keep on providing every year these new fly strains whenever they want it. Now, some of the things that we can learn from Drosophila. One is mitotic chromosomes. Typically, one we used to do from mouse bone marrow, But now that mouse dissection is not easy, this simple thing to look at mitosis has also stopped. But Drosophila provides a simple way to do this. All that you need to do is take healthy 30 cell larvae, dissect them out, take out these brain ganglia, fix them, give them before fixing a small hypotonic treatment, fix them, dissolve them with 50% acetic acid, and make these beautiful air dry preparations, which one can stain with GIMSA or SEEN or anything, and look at microscope and look at this. And these chromosomes show you the features of heterochromatin, euchromatin. Many of these things can be studied. Then, of course, the most remarkable thing Drosophila offers is these polytrin chromosomes, which are dream of cytology and geneticists. And these are again very easy to prepare. You again dissect larva, take these larva glands, fix them, and squash them. And then you see these beautiful chromosomes, which, as I'll show you later on, have been instrumental in letting the development of molecular biology, in fact. All our understanding of gene regulation and gene ovulation, all the studies had roots in studies on polytrin chromosomes. They show these very large because they became endoreplicated, and because of which, and the chromatin remains less coiled. So you see each chromomere of different sister chromatid that are produced as a result of endoreplication remain paired, and therefore we see them at dark bands and interbands. And of course we see these puffs which are visible as swellings. So these have been something very remarkable thing and very simple to prepare. Now this is this paper I would want everybody to appreciate. Paper in 1934 by T.S. Penter, a postdoc of Morgan, and who first identified these are polytene or multi-strand. Poly means many, T means threads, that many threads make these chromosomes. And in that paper, the editor wrote this. Ever since, to introduce that paper, ever since the gene hypothesis was generally accepted, now mind that this is way back in 1934, geneticists and cytologists had been dreaming about something, and this is where they saw that they now can actually see genes, not just to think in terms of locus on a hypothetical genetic map. Now they could be seen that yes, each of them is a gene, and actually physical mapping of genes in Drosophila started way back in 1934. And this is what the conclusion by T.S. Penter in that paper was. With these four discoveries before us, that is, constant and distinctive patterns, somatic synapses, the behavior of active and inactive regions, and the separation of arms of the large autosomes. Mind it here, 1934, they didn't know what genes are, they didn't know what gene activity means, but they had imagined this, that the puffing is the active region and the bands are inactive region. Now this is remarkable farsightedness, but that's what it was. And then he says, it was clear that we had within our grasp the material of which everyone had been dreaming. We found ourselves out of the woods and upon a plainly marked highway with bypaths stretching in every direction. It was clear that the highway led to the layer of the gene. Layer is where, in English language, layer means the place where lion lives. So the comparing gene with lion, but now we could reach that lion and we could attack the gene. The paper of the title is Polytrin Chromosomes and the Attack on Gene. Now this is very interesting. And what is stated here has actually happened in within 3-4 decades. All these points were realized. So this is the map that Brussels had drawn in 1934 for the exosome. These were painstaking hand drawings, not photographs. And I'll show you that details that he had drawn are really unbelievable. Using his simple microscope and spending months and months on microscope and his drawing board, he drew every band that was visible. And the precision of his every band was, for example, if you look at this map in this line here, where you see these drawings, hand drawings of by Bridges, and this is what under electron microscope these chromosomes look like. And every dense band that Bridges had drawn is visible in electron microscope. The important point is, electron microscope did not discover any band that Bridges had not seen with light microscope. Now this is unbelievable, and that simply reflects the power of observation. And I think if you develop that kind of perseverance, power of observations, we don't have to always worry about high-end equipments. Not that high-end equipments are not necessary, but they are not the only thing by which we can make great discoveries. And this map of Bruza is drawn in 1930s. Till date remains absolute gold standard. No modifications have been needed in this map, and we work with Drosophila, melanogaster, chromosomes and genes, refer to this map every time. And now what has happened is with molecular biology, each of these bands has been mapped in terms of DNA sequence. The entire genome of Drosophila is sequenced, and so now we know from which band, which base number to which base number, with this band 1 and band 1A1, 1A2, 1A3, and so on and so forth. This has been precisely mapped. And in addition, specific genes were mapped way back in 1930s as to which band the gene belongs. Why could this be done? Because now we can see these chromosomes, these dark bands, and I would say that these are really, you're looking at genes directly. And so what have been some firsts with polyploid chromosomes? That gene activity requires losing of chromatin. We now know that the nucleosomes are reopened up and so on. But this was something when puffing was discovered in 1930s and 40s and early 50s. Bierman explained that puffing is chromatin loosens, and that shows chromosome opening. And this happened before we knew that genes are made up of DNA. This phenomenon was predicted long back, that there is a fiber which opens up and that becomes active. And of course, physical map, which in humans have been very important, but in case of Drosophila, physical maps were made as early as 1930s. Another important point that polyploid chromosomes and Drosophila studies showed was the steroid hormones act on genes and induce cascading pattern of gene activity. All the steroids that we talk about in humans, estrogen, progesterone, testosterone, that they act directly on genes was discovered in Drosophila polyploid chromosomes first by directly exposing to exosome, and then see that you see a cascade of gene activity. I'll show that later on. And then the whole process of heat shock induced gene activity was discovered in Drosophila, because here you could see actual puffing change. I'll show this again later on. Then when a gene becomes active, now you have possibility of looking at that when a gene becomes active, how does chromatin change, what kind of molecules are associate there, what bring about what regulatory molecules, transcription factors, and bind there, and how the transcription machinery is activated. All this can be seen with these chromosomes. For example, physical mapping. See, because the homologs are paired, so if you have a small duplication, theoretically we draw it like, okay, deletion, that gene 3 and 4 are missing in this homolog and they are present in this. And we can see in polyploid chromosomes, we concentrate on this point, that you have these bands which are missing on this homolog. And so immediately you can make out. Now, using this kind of technology, we can even map a promoter, as we had done way back in 1996, that we mapped the promoter by using small deletions at chromosome level, that when a particular region was deleted, the gene could not be activated. And so polyploid chromosomes have provided enormous information. And I think looking at this on a microscope can really mesmerize students. They can enjoy looking at genes. And this is what we can see. For example, if you take late 30s larvae of different age and you prepare surveillance on them every 1 hour after, you can see the dramatic changes in puffing pattern. This study, which was done by Michael Ashburn in the 1960s, really established that, for example, this is pre-ecdysone. That this particular first panel A is before ecdysone is given, and then as the larva is releasing ecdysone, or if you do it in vitro, you expose the surgeon to ecdysone and then withdraw ecdysone, and then you can see every hour these changes. For example, this was condensed here, condensed, it opens up much more and more, and then goes back, regresses. So we can see the dramatic change in gene activity, which we cannot see in any other cell, because we can't see the genes when they are active in there. But polyploid chromosomes provide that, and that's the advantage that I think we should make use of. The discovery of heat shock response was again an accident in Fiducia Ritosa's lab in 1962, that some survivors from some larvae exhibited unusual puffing patterns. And then he discovered that it was because the larvae were exposed to elevated temperature. And this is what we can see here. This, for example, from a normally grown larva, polyploid chromosome, this is from a heat shock larva. Now if you concentrate on these 87C here, they're completely opened up, but in this control larva, these are dark bands. And you can see this change within 5 minutes, that you take surveillance from a normal grown larva, expose it to 37 degrees centigrade for 5 minutes, or take a larva and expose larva to 5-10 minutes, and make chromosome progression, you can see these changes, these new puffs. And that's what they're called as heat shock puffs. Later on, of course, they could be shown that associated with these heat shock puffs are synthesis of the new heat shock proteins. And so this opened up a whole field of stress biology, starting with these polyclinic chromosomes. And this can be done in any classroom. Only thing is, you will have to ensure that your control larvae remain at 24. If they are exposed to 30 degree or 32 degree even for few minutes, already heat shock puffs will become active. So that's one limitation here, that you need a controlled temperature room. The heat shock response can also be seen now today thanks to transgenic lines. For example, this particular line that we have also used and has been made long back makes use of the HSP70 genes promoter fused to beta-glucosidase gene, LAGG gene of E. coli. So now what happens is, when these larvae, this is the gut of Drosophila larva, these are the malpighian tubules, when they are exposed to elevated temperature, the HSP70 promoter drives beta-glucosidase of E. coli, the LAGG gene of E. coli, and so beta-glucosidase is synthesized, which can be detected by histochemical staining using XCal as the substrate, which is yellow colored powder, but when acted upon by beta-glucosidase, turns into magenta blue. And so the whole tissue, every tissue of Drosophila expresses heat shock genes, and this can be seen very, very conveniently. The other area which Drosophila has really revolutionized is developmental biology. And this is a very interesting thing. To begin with, Drosophila embryology looked very bizarre, completely different from what we have learned about previous divisions in frog or Amphioxus or mouse or sea urchin. Here, the initial zygote keeps on dividing nucleus without cell division. So you get a syncytial mass of embryo with thousands of nuclei and common cytoplasm. And then at a particular stage, which is equivalent to blastoderm stage, these nuclei migrate to periphery. Mind it, this is still a single cell, and these are independent nuclei aligned at the periphery, and then now the cells form. So the cleavage is, the actual cell division is delayed much more. After about 13-14 cycles of cell division, when you have thousands of nuclei in a common cytoplasm, you get this. And then there is no typical gastrulation. There is something called, equal to that, is germ bind extension. But so this was very unusual. But now what we know is this unusual development has become a real advantage in learning how genes determine development. How genes determine that this should be head region, this should be thorax region, this should be abdominal region. This is what was done by using simple genetic tricks. And so this has been very simple way. And now we can look at these processes, there are simpler methods to look at these in any classroom, that how this development happens. For example, this is a simple thing which has been created by Dr. Shamla and Masula. They teach it, that you can collect eggs over a period of time in this petri dish with agar, using this kind of a bottle. You can make it at home and have these flies lay eggs on this. This is, for example, the petri dish containing these numerous white dots, which are the eggs. And you can make a egg filtration device like this. Take any plastic bottle with a screw cap, remove the central part of the cap, but so the rim can be still tightened, and put any fine cloth under this, nylon cloth or cotton cloth, clean, and tighten it so that the cloth will hold the eggs on its mesh. And now you can treat each of these eggs, pass them through the different solutions without worry about losing. All that you need to do is, the egg is covered with a chorion layer. For example, these are the two chorion filaments, and the whole egg is covered with chorion layer. So that chorion layer, which is an opaque white layer, has to be removed, which can be done with simple bleaching powder. All you need is a 5% solution of bleaching powder. Treat it for a few minutes, and the chorion dissolves, wash away the bleaching, and now we have embryos which you can examine under even bright field. Your phase contrast is much easier, but even if you look in bright field, you can see these kinds of developmental events happening. The other thing that one can do is, if one has a fluorescence microscope, one can stain the nuclei in this with DAPI fluorescence stain, which can get into live cells, and then you can see the mitotic divisions and the nuclei very clearly all through. And this kind of experiments can be done in the classroom where students can see how the development happens. And what one can also notice is at the posterior end here, one can see the first cells that are formed are the pole cells, and they are the exclusive precursors of germ cells. So one can, one can see the difference between soma and germline if one does this kind of experiments. The other thing that we can look in developmental biology in Drosophila is how the adult organs are formed, adult structure, legs, wings, and so on, which larva doesn't have. And what we know is that within the body of the larva, we have these imaginary discs. These are embryos. These are the real discs that one can see in the microscope. They are located around the brain region. This is the larval brain, the brain ganglia, and this is the ventral nerve cord. And these are the different discs. Each one of them will give rise to an external structure. And for example, this particular we call wing disc, which has a shape like this. This part will become the wing itself. This will form the junction with thorax. This is the halter disc or the leg disc. This will form the antenna part. This will form the eye part. And so we can look at this. If we take very early pupa and dissect them, we can see for example, this is a developing leg, so segments are now forming. To begin with, it was like this, but then as it develops, it begins to make these segments. So one can do lots of experiments with these. Lot of genetic studies have been done to find out how does a cell know what it has to form and so on and so forth. But the other thing is that morphologically, one can do these experiments in a very simple way. The other thing that Drosophila allows is the mutation, genetic mutation. E.B. Lewis, for example, who had worked whole of his life on this bithorax phenotype. That means normally we have only one pair of wings. That's why we call them diptera insects. The second pair is present as a very small halter. Because of this mutation in the bithorax gene, the second pair also develops as a full leg. And that's what Lewis examined, that what are the genes, what interactions, and he discovered these homeotic genes, and that's what led to the understanding of the Hox homeobox homeotic gene. That these genes, it's not in Drosophila, for example, we know it from bithorax, but in humans also same gene is present. Actually, the human gene will do same function in Drosophila. If you put Drosophila gene in mouse, it will do the same function, although they give us different structures. Because what we now know is what these genes are telling the cell is its coordinates in the body, whether it's in proximal part, distal part, and thereby regulating downstream genes. And this has been a remarkable discovery. Another mutation like this that we call homeotic mutation, which are discovered way back in 1949, is that you have this antenna, which because of the antenna PD mutation, antenna develops like first leg. Obviously, this fly will be a great research model because it doesn't have an antenna, but for geneticists, for molecular biologists, it becomes a very important way to understand what makes an organ develop into what it is. And one can look at this. And this is what the Musland Follard and colleagues, Eric Wishaus, again did a very simple genetic study, induced mutations in wild type Drosophila, look at embryo that die as embryo, and then examine why did they die. And they discovered these homeotic genes, the gap genes, the pair rule genes, and the segment polarity genes, and which even today we can see. This is very simple. You don't need any sophisticated equipment. All you need to do is look at the larva or embryo under microscope, and you can see these segments, the dentical patterns. If a dark field, these images are dark field microscope, because then the dentical patterns, which are highly dense, appear bright. And so one can easily make out that, okay, there is something missing here. That means the gene that are mutated are responsible for making this region. These were part of a gap gene mutation. And so you can have the different mutants and examine them. And of course, E.B. Lewis, Eric Wishaus, and Musland Follard got the Nobel Prize in 1994. And actually, I wrote a commentary on this, 1995, that this was a tribute to power of genetics. These Nobel Prizes didn't employ any molecular biology, any sophisticated equipment, simple plain genetic mutations and examination of the eggs, examination of the adult flies, and how do they behave. And that's what led to their Nobel Prize winning discovery. And the important point is that any classroom can look at these phenotypes in a simple way with basic microscope and basic stereo binocular things. It doesn't need any sophisticated things. Likewise, we can induce mutations, we can cause a misexpression of a given gene, and then we can see that instead of this naked cuticle here between the segments, one can get these forming everywhere. So one can understand how the genes are regulated, how they break the cells to make what they do. We can also look at, again, using beta-glucuronide reporter gene, LAG-Z constructs, where you different promoters and put beta-glucuronidase downstream of it, make a transgenic fly, and then examine where beta-glucuronidase is expressed, so that without doing molecular biology, we can exactly know where the gene is expressing. And some of these can be done in classroom without any great difficulty. The other experiment that we can do is that exdysome controls preparation. A simple experiment. You take a larva, take a fine thread, tie a knot around somewhere, and then see the larva is amputated. Why? Because as you have tightened it, the exdysome, which is secreted from prothoracic gland located above brain region, doesn't move to the body, and therefore these cannot undergo molting. And so this is again a simple experiment, and we can do it in Drosophila. No fear of animal ethics or cruelty on animals and so on, fortunately. We can do molecular biology, we can extract DNA from flies, we can look at the DNA in the standard way, that the same protocol as we do for mouse or anything else. All you need is a centrifuge, and that's it, and flies. And then agrogel, so they can see the DNA, the marker, and the intact genomic DNA, shared DNA, or you can digest and can see different fragments. So these are again Drosophila flies you can get in large number. Now there is a protocol that we can isolate DNA from a single fly and examine it. And that's where the sensitivity has become very useful, that a lot of molecular studies can be done with this kind of technique. We can look at if there is any mammary toxicity, xenobiotics are there or something else is damaging. One simple way is we can measure the fecundity of male, whether males are affected, females are affected. We can simply count in a given unit of time per fly how many eggs are laid, how many eggs are hatching out. And again, these experiments don't need, all you need is a way to grow flies and treat them through the food or by aerosol to some xenobiotic, and then see how it affects the fecundity of the fly. Again, simple experiments. The only thing is it can't be done in a single day. It will need 2-3 days time to look at, do the treatment and examine the progeny and count the eggs and see how many eggs hatch, how many eggs die, so that one can make out. So it can be very good assay for environmental toxicity or any genetic toxicity, any genetic factor. This can also be used to identify genes that control fecundity. If there's a mutation and if that reduces fecundity, then we know that there's doing something with the spontogenesis or oogenesis. Other way for immunity study, Drosophila doesn't have adaptive immunity like the antibodies that we have, or T cell or B cell, but they have the basic innate immunity. And that basic innate immunity is common in Drosophila as in humans. And in this case, there are a number of small molecules that act as defense against specific testing. For example, lipopolysaccharide is a common antigen that can be used in Drosophila, and it will elicit response of certain genes. For example, sarcopene is one of the innate immune response gene in Drosophila. And so if we have a transgenic line, sarcopene A promoter fused to LAG-Z of E. coli and a transgenic fly. Now, if these larvae carrying this are exposed to, let's say, LPS or infected bacteria, in the Malpighian tubules, which is one of the immune organs and homolog of human kidney, but it also is an immune organ in Drosophila and insects. And you can see synthesis of appears a blue color, which indicate that the sarcopene gene has become active in response to infection, and so one can see the immune response being mounted. And what stage it mounts, when they are weak, when they are less. So if you do the same thing at first instar, response is very weak because first star still is not ready with its immune response. But as it matures, the immune response becomes better. Then another interesting thing is the behavioral genetics. The whole behavioral genetics, field of behavioral genetics started when Sunil Benzer, who many of us know as the person who did the R2 locus fine structure of gene, who gave the term cistron, recon, muton. Just before I go about Sunil Benzer, something more interesting. In 1940, Sunil Benzer was involved in making transistors. He was a physicist. In 1949, he turned into a fly geneticist. Straight from physics, he jumped to fly genetics because he heard a lecture of Sir Max Delbruck, and that made him convert himself into a biologist. And then he looked at the R2 locus of T4 bacteriophage for about 10 years and came up with a brilliant explanation of recon, cistron, muton, and so on. And after having done that, he decided to jump to flies. And then he asked this question. There's an article available on him that how he did it. He started looking at human behavior and saw that, okay, even his two daughters were twins, behaved in different ways. So his question arose that why should they be different? And he started using Drosophila behavior. So he became the father of behavioral genetics and using Drosophila as the first source. And till date, Drosophila remains a very good model for neurogenetics or behavioral genetics. And Obaid Siddiqui is sitting somewhere here. He learned, we set up the TFR fly lab, his behavioral genetics from Sunil Benzer when we visited him as a postdoc. And that's where the behavioral genetics started. And you see what is interesting is the fly can decide whether it should jump from this point to this point or should fly. It can do that estimation. And this is very interesting to think of. One simple way one can look at Drosophila without needing any fancy things is courtship behavior. So all that you need to put a mature female and a male in a petri dish and allow them to acclimatize. In 5 minutes, then you can see that the female is not already mated. It should be virgin female, but 4 or 5 days old virgin female. Put it with a male, and then within few minutes, you can see that they start this stereotype behavior. Absolutely. But if there's a mutation, then something goes wrong. They don't behave like this. Or if the female is mated, it will not respond to the male's approaches. And so this becomes a very good way of looking at the behavior. For example, the lower panel showed the actual videographs, the photomicrographs of things happening inside a petri dish. You don't need any sophisticated equipment, just a small glass petri dish. One can look at how larvae move, whether their musculature is affected, because several diseases, neurodegeneration diseases, nerve diseases, or certain treatments affect their motor functions, affect their muscle contractility. So one can simply track, take images using even smartphones. One can take a small video and see how the larva move from here to here, here to here, or it remains moving only in a small area, and then one can do an image analysis using simple statistical tools to find out whether it's moving freely or it's moving in a restricted area. And this can give us an idea about the muscle contractility, the regulation of the muscle movements, and so any defect in muscles, any defect in neurotransmission, any defect in brain function can be directly assessed by this. And these are very interesting experiments for students to do in classroom. Similarly, we can look at that flies are typically negatively J-tactic. That means they move away from center of gravity. So if you put flies in a tube, and wild-type flies will tend to move towards the upside. But if the motor functions are affected, for example, one can treat the flies with rotenone, which is one of the insecticide, and because it damages their neurotransmission, then the fly don't move up, they remain bottom. So all you need is simple test tubes or even a 10 ml pipette where you can mark a distance or how much time the flies take from, you tap them down and then let them move, how fast they can come up to this. The ones which have compromised muscle movement, neurotransmission will not move up, and this can be a very good way to assess the behavioral part of it. One can look at attractant and repellent smells, because for insects, smells are very important. Olfactory sensation is very critical. And so all that one can do is one can put larvae in the center here and on one side put a cotton bag with one volatile component versus another volatile component, and they see if larvae move towards one way or the other. If they don't move toward this side, that means this chemical is repellent. If they move more toward this side, this is attractant. And so this is again a simple experiment which doesn't need any fancy equipment. All you need is just a small petri dish and some of these chemicals, which can be used to look at the behavior. The Drosophila eye is a beautiful thing to look at. It's a compound eye with almost 700 eyes in each one. Now this is something interesting. We have each eye has one lens. Here you have each fly has two eyes, each eye with about 700 to 800 lenses. How does it coordinate its vision? It's another interesting thing. But the thing is that we can see this beautiful pattern. Earlier people used to do this with scanning microscope. This image would be generated by scanning microscope. But an excellent discovery in our own lab by one of my students, Richari, who is now my colleague, she developed this nail polish imprint technique. All you need to do is take the adult fly, remove its head, cut its head. You don't need to do any fixation or anything. Just put a small drop of transparent nail polish on its head. Wait for a few minutes, and then using fine needles, crack open this nail polish which had dried up, and carefully remove the nail polish layer over one of the eyes, and go and see in the microscope, and you get a beautiful imprint of each ommatidium. It is there. You can see in higher magnification, and you see same resolution as one would get with scanning microscope. And this simple technique, it needs just 5 minutes. All it needs is some flies, transparent nail polish, slide in a cowslet. All you do is you take this nail polish layer, put on a clean slide, put a drop of 50% glycerol, mount the cowslet, go to microscope, bright field or phase or DIC, and you can see these patterns, beautiful arrays. If there is any disruption anywhere, any gene that has gone wrong, or there is any neurodegeneration or something that you treated the flies which affect the development, you can immediately see this disruption, as I'll show you. And so this model has been extensively used to study human neurodegeneration in Drosophila. We have models of Huntington's, Alzheimer's, Parkinson's, where the scar or the spinocerebellar ataxias in Drosophila. We have the mutant human genes put in Drosophila, and we can activate them when we want, using again a very interesting system of having one fly which expresses a protein called GAL4. This protein is from yeast, and this is put under a promoter called GMR. GMR promoter is expressing only developing ibis. That's not very specific. And so this fly will make this GAL4 protein in its developing eyes. But this protein is a transcription factor. There's no target in Drosophila cells, and therefore it remains without any consequence. We have another transgenic lines in which we take the US upstream activity sequence again from yeast with which GAL4 binds. This is a transcription factor binding site. We take this sequence upstream of any gene that we want to express. For example, here written polyQ, that is a mutant gene that has a long stretch of mutant residues, as happens in Huntington's, for example. So when we cross them, then in this progeny fly now, this GMR will make the GAL4 protein made in this part of the eye disc, where the eye is developing, ommatidia are forming here, and in these cells, GAL4 will bind to the US sequence and cause expression of this polyQ protein. If this polyQ protein is toxic, you get degenerated eyes. If it is non-toxic, you get normal eyes. So one can look at this. So any gene that affects eye development causes neurodegeneration, it becomes a simple assay. You don't need any molecular biological things. All you need is these flies, cross them, and look at this thing. And now, to look at this, we can use again the nail polish imprint technique. But if the arrays are normal or mutated, we can very clearly see them. And so it becomes a very simple way to look at if there is a neurodegeneration happening. So eye model has become, the Drosophila eye has become a wonderful model across the world for looking at various diseases that affect neural development and differentiation. And this can be done again in classroom without any fancy things being required. So what I tried to do here is given several examples. There are many more examples that Drosophila can be used, although it was initially used for studying in genetics, and that's what people still think that it's used only for genetic experiments. But now it's time that it can be expanded to many areas in biology. And to facilitate this, we are now have compiled this book, which will be an e-book freely available across to anyone from Indian Academy site. We hope that this will be available by end of this month on the web page. It's about 800 pages or so of e-book, no hard copy, and where each of these different practicals, about 85 classroom practicals are described, which can be done. Some of them, of course, need a little more sophisticated facilities, but a large number we have thought should be, which can be done by any laboratory which has some basic facilities, and which can, and of course, as I said in the beginning, that there will be ways where these flies can be obtained by the desiring colleges for doing their experiments, classroom. We don't maintain it over the years. If they can maintain, very good. But if they cannot, then the flies can be obtained from some of the stock centers or labs where they are being worked, and so these students can be exposed to them. So with that, I'll stop and take questions. I hope this has been useful in some ways. So I'm now ready to take any question that you want.
Thank you, Professor Lakhotia, for this excellent session. We have gone through the exciting journey of development of Drosophila genetics from the very beginning, from the first physical map to the developmental, behavioral, and neurogenetic aspects. Thank you once again. So, shall we take the question right now, sir?
Yes, I'll be happy to answer.
Okay, participants, you can unmute yourself and ask questions to Professor. And in the meantime, let me read out the questions we have in the Google chat box. So, there is a question, sir, how to screen anti-diabetic activity in Drosophila?
Okay, that may not happen because there are models for diabetic studies that you can feed them on high glucose diet, create diabetic conditions, and then see what kind of treatments can reduce their thing. I'm not fully aware, but I know one person who is working in Lucknow, Dr. Naveen Gautam. He is using diabetic. There are some other people also who are using diabetic model in Drosophila, and this can be done. It has become a very good model now.
Thank you, sir. So the next question, how do we run Western blot analysis with Drosophila body?
Well, Western blot, you see the protocol remains the same thing. They you homogenize, take proteins, run them on gel, separate the proteins, and then transfer the proteins through as you do for either by simple diffusion or through electroblotting, and then use the specific antibody that you want to do. This ebook that we have will have a protocol on this also, a detailed protocol how to do Western blotting with proteins from Drosophila.
Okay. So which model of organism we should use and why?
Well, each model has its own advantage. Each model will have certain things that it offers, other models do not offer, and certain things that we discover in one model may or may not be applicable in other models or human beings if we are trying to look at it from human point of view. But then also depends on where one is trained, what are easily growable, which can be maintained easily, and so that's what should be done. So I won't say that this model is better than other, but then the advantage of Drosophila is the numerous thousands of mutants that are available, transgenic that are available, and the remarkable thing that Drosophila community offers is free sharing of material. a tradition that's started with Morgan, that all the mutants that are available are freely shared. Even newly discovered mutants, we all share with people without any IPR issues or patents issues or something. And the stock centers which are there, unfortunately in India we still don't have a typical stock center, but there are certain labs which will be willing to share the flies as you need it. And so from that point of view, Drosophila remains a very good model.
The next question we have, is there any gene for aging in Drosophila, and what happens if we remove that or those genes?
Well, the point that we should understand, there is not one single gene that causes aging. Aging is a complex biological process, and there will be a large number of genes involved. And that they have a lot of research in aging, but then it's a kind of quantitative character, a variety of factors. And so we can't just remove one gene. We can have different genes because people are selected for lines that are long-lived flies, people are selected for lines that are short-lived flies, but they don't find a single gene mutation. That is a complex thing. But Drosophila is again a good model for aging studies because one can look at the change in brain, one can look at changes in metabolism, one can look at changes in the reproductive system, and so it has remained a good model. But I don't say that we can identify any single gene that this is the gene for aging. And for example, we also show that some of the Ayurvedic formulation like made of Amla can prolong life in flies also, as they have been trained in humans. So this is one area where we can test for effect on the life quality as well. That Drosophila can be used for that.
Next question, what is transgenic fly?
Well, transgene is actually a gene which has a DNA which is different from the host's own DNA. I mean, from that particular species DNA. So basically we take DNA from some other organism, manipulate it in vitro by genetic engineering as we wanted, we create a sequence, and then introduce that into the host genome. Where we want to do, and that becomes a transgene, and that organism is called transgenic. So if I say transgenic Drosophila, that means this Drosophila has its own genome plus something else that we have introduced in it deliberately by our choice. A gene taken from humans, a gene taken from another Drosophila species, a gene taken from bacteria or a plant or any other organism. That will be a transgene.
Okay, so next question we have, sir, how do we distinguish between male and female Drosophila? Is it depend on size, like the female one will be a bit large in size?
Well, male and female can be distinguished. First is size. Yes, females are larger than males, but then there are very distinct morphological differences. The posterior end of a female is more pointed, male is rounded. And male in Drosophila melanogaster, the tip of the abdomen is much darker, blackish in color, whereas in female it's not that color. There is no black color, no black melanin pigment there. Another important point between male and female is the presence of what is known as sex comb on the one of the tarsal segments of the first pair of legs, which a pair of stout bristles which are present only in males but not in females. So that's the way we distinguish between male and female easily.
Okay, next question. Sir, what could possibly be the problem if fungal contamination, I think it's contamination, occurs for a transgenic line of Drosophila, but not when the same medium is used for native strain?
Well, I don't think that transgenic strains are more sensitive to fungal infection. No, that's not true. Because fungal infection not coming because of the transgene. Fungal infection is coming because there's something else is contaminating. So it's not that only transgenic lines will have fungal infection and the others will not have. They can have, and that's where fungal infection are rather difficult. We have a series of processes to be done, that every generation we quickly change the flies to new vials and new bottles, and keep on treating them with something. Actually, in the Drosophila food, we add nifedgine as an antifungal agent, which normally keeps fungus away. But if the fungal infection is strong, then we need to do some treatment. But it's not that transgenic lines only are sensitive to fungal infection. That's not true.
Okay. So, sir, how to count Drosophila neuron by flow cytometry?
Well, flow cytometry can be used for anything, counting any number of cells. But the way neurons can be counted by flow cytometry, there are certain lines available in which neurons will be expressing GFP, reinforcement protein, and there will be fluorescence. Or we can have different neurons only expressing GFP. So we can use such transgenic lines where we have GFP expression happening in either all neurons or a certain subset of neurons, and then we can make a suspension of brain cells from large number of lines, pass through the flow cytometer, select for GFP expressing lines. So these are the ways, but they are not absolutely 100% correct. There can be always contamination, and you need a large number of flies to do with. All the people have tried with fewer numbers, with much more sensitive flow cytometric system, and that is to some extent possible. But you need a marker, and that marker provided in a transgenic system, whereby we express GFP or some other fluorescent marker in those specific cells.
The next question, so which method we can follow to isolate DNA from Drosophila?
For extraction of DNA, it's the same method.
Yes.
Homogenized in a SKS containing solution, and then the ethanol, the phenol chloroform extraction and ethanol precipitation. The process is absolutely same as one would do for any plant material or any mammalian material or fish material or bacterial material. Only in plants, of course, you have the problem of removing cellulose in the first place, but in these cases, you don't need anything. All you need is SDS, and then phenol chloroform extraction, and finally precipitation with ethanol. Same method.
Okay. Sir, what is the process to make a stock of CRISPR mutants in Drosophila?
Well, CRISPR mutants will require again a complex system. We need a variety of lines. Many of them are available now that they will express the Cas enzyme, CRISPR Cas enzyme. You need to create your transgenic with the guide RNA, and then cross them so that with the Galfo driver, so that the CRISPR Cas becomes active in germ cells, and then it will do its own. So these stem lines are available. All you need to do is generate the RNA and then make a transgenic that will express that particular guide RNA. And fortunately for this, now there are services available. For example, CCamp in Bangalore has a fly facility that can generate CRISPR-Cas lines for you on payment, which is not very cheap. It is a little expensive, but they can do it.
Okay. In human or animal, a cell dies after few division due to programmed cell death. But in case of amoeba, what it does happen, and they are called immortal.
Well, partly I think there's a big question, very very fundamental question, that why amoeba doesn't die. But part of the reason is that amoeba doesn't have typical sexual reproduction. The organ that introduced sexual reproduction introduced a limited life. And why biological system have to do that kind of system? But that is what we look in every organ. Many plants never die. You can have Israel propagation; they can continue. But the moment we come to sexual reproduction, there is a limited life, and that's partly because of the regulated form and function that we have. See, the price on your pages, amoeba doesn't have a face like us. We have a defined body shape, amoeba doesn't have. But that's because it just keeps on dividing. Of course, one can say paramecium has a definite shape, and it can also keep on dividing. But paramecium, after a certain number of cellular reproduction, will not be able to maintain itself unless it undergoes through a conjugation cycle. So these are basic fundamental biological processes. which we partly understand, partly we still do not understand as to what makes. But then what we know is that now all our system, telomere is one of the important points to be, which defines how many times a cell can divide. So telomere maintenance becomes an additional issue that one has to take care in those who have finite life or cells that have finite division capacity.
So, sir, there are a few more questions. Shall we take?
I'm okay. If your time
no, it's not about our time. If you are okay, then we can proceed. So the next question, sir, what is the effect of Amla in Drosophila and human gene or genes?
Well, what we looked at was Amla Kirasan, a very simple preparation, a standard Ayurvedic preparation, and that we saw that it does prolong life of flies. It makes them more heat shock resistant, oxidative stress resistant, as was expected. But more importantly, what we found was that it suppresses Huntington's symptoms in Drosophila models. Many polyQ models we checked that Huntington's or some of the spinocerebellar ataxias, and when these larvae were fed on Amla Kirasan, where we were expressing these genes by our genetic program, and there were neurodegeneration, but when we fed them on this, neurodegeneration was substantially reduced. Not completely gone, but substantially reduced. And this we subsequently, after doing this, we also looked in rats in collaboration with CCMB, and there too it does same similar thing. So Amla is certainly, I would say, a good medicine for brain and for life in general, as long as it is prepared in a careful manner. When I mention this, I should also caution people that many of the commercial products that come in name of Champras, different so on and so forth, are not properly prepared and can actually be harmful than beneficial. But a well-prepared things of Amla will be very good. One can take even raw Amla without having all these fancy preparations. That cannot be equally good. And that's what we learned from Drosophila again. We provided the evidence that yes, it does. And the mechanism. We could look at what kind of genes are activated, what kind of genes are not activated, and why should it provide that advantage. These were published already.
Okay, so next is how Drosophila study contributes in cancer genetics.
Translational genetics.
Cancer genetics.
Well, we have models of cancer. Like many labs, a large number of labs are working on cancer using Drosophila model. The advantage in Drosophila again is we can induce cancer when we want. See, one can induce cancer in Drosophila also by treating with something, but then there is a more important is that you can genetically program that at this particular moment a cancer cell will be generated. There are genetic tricks that we can do that. And that's what we are also utilizing, that we create clones of cancer cells, and then we can follow their progression. We can follow what kind of treatments will reduce them, what kind of things that can happen. So Drosophila has been a great model, and in fact, historically, it was only study on Drosophila that gave the concept of oncogenes and the oncogene suppressor genes, chemosuppressor genes. It's because of studies in Drosophila that this concept started. And so Drosophila remains a very good model, and as we learn more about it, as we develop more kinds of transgenic lines and other things, it becomes more and more attractive.
Okay, next is, sir, is there any method for the cryopreservation of Drosophila embryos with promising viability?
No, so far cryogenic preservation has not succeeded. There were major projects taken up in last century, but none of them succeeded. That's one limitation, that we cannot cryopreserve flies or embryos. So far, no. There's no technique available.
Okay. So the last one, can we conduct any medicinal tests in Drosophila which we conduct in human? Will be the results are same or not?
Well, what kind of medicinal test? I mean, you see, if you are doing some biochemical tests like estimating sugar, estimating fat, and then the techniques remain same, no matter whether it's the sample comes from human or from this thing. But to diagnose a human, we need a human to be tested. But then the studies can be done. We can simulate parallel studies in the Drosophila flies and then gain information and then make their use with certain clear understanding in humans as well. That's where the fly models become important. That we have fly models for several heart diseases, we have fly model for obesity, fly model for diabetes, fly model for a large variety of neural disorders, muscle disorders, and so on and so forth. The models are available. So one can do that kind of study in them and then see how much of it can be applicable to humans.
Okay. Is there any other question? Participants, you can please unmute yourself to ask question. I think there is no more question. Salma, is there any question in YouTube Live?
So there is no questions.
Okay. So let me thank our honorable speaker for this excellent session once again. Thank you, Professor Lakhotia.
I'm very happy that you people are doing this kind of activity. Keep doing well. And anybody can feel free in writing to me anything that we can help. It will be our pleasure. Do well.
Okay, that is great. Thank you, sir. Thank you once again. So we are concluding the session, and dear participants, please follow the WhatsApp group for tomorrow's lecture and all other details.
Okay,
So thank you everyone.
Bye.
Bye bye.